In Lab 10, we are going to cover basically an introduction to the muscular system, and then we'll review our muscle histology and the 3 types that we have previously talked about. Most of today we are going to spend on skeletal muscle, so we've kind of gone over a basic introduction to skeletal muscle, but there are a lot more weird things to look at in the histology. We're going to talk about the unit of the muscle cell and kind of all those things we find in there. And while a lot of that is not going to be tested on lab exam 2, and I will specify what I want you to focus on, all of this is super good information to know just from a health standpoint if you want to be educated about what you're doing during a workout with resistance training. This is all kind of the behind the scenes information that goes on to increase muscle strength and all that good stuff. And then a lot of this is also fair game for lecture. So we're going to cover probably a little more detail than you will be tested on in lab, but it's all good stuff to know. So let us start by talking about our 3 types of muscle. And so a good review for this would be to look at your lab manual exercise 4 with histology and just review that information about smooth, cardiac, and skeletal muscle. So briefly, we're going to use the Yale Histology website and then the Protein Atlas website to review some of that tissue. And one good thing to know is that we are now getting more organ system specific. So far we've looked at these isolated images of muscle histology. Now what's going to happen as we move through the organ systems is you're going to see examples of all of these types of muscles in the heart system, the cardiac system. We're going to talk about the digestive tract, where we see a lot of types of smooth muscle. And then, of course, we'll have our upcoming muscle unit. So let's go ahead, and I've got them pulled up here. We're going to look first at our protein atlas and start with smooth and heart muscle. So this should be good review. I do recommend using the human protein atlas for cardiac, smooth, and skeletal muscle. So we are looking at the cardiac system right now. This is a histology slide from the heart. And we've all seen this tissue before, hopefully, because it is going to be on lab exam 1. But what we see is we've got a striated muscle tissue. We'll talk about this, but cardiac muscle and skeletal muscle do have these striations in common. Those striations are caused by something called a sarcomere. So that's coming up in just a few minutes. However, cardiac muscle is involuntary. So that's the main difference. And then we have one additional feature here called an intercalated disc. So let's zoom in just a little bit if we can. So basically our intercalated discs are going to be these kind of perpendicular lines running through the pattern of muscle that we see in cardiac muscle. And these are sometimes called gap junctions because they are helping electrical signals to bridge the gap between muscle fibers. And so when your heart contracts, we have this electrical signal that has to propagate all the way through the muscle fiber so it contracts in sync, and that is aided by these intercalated discs. The other main thing we see here is this branching fiber pattern. So skeletal muscle, as we've seen, has a really regular pattern of striations, whereas cardiac tissue is going to resemble more of that branching bamboo pattern. So that is cardiac. And then we have, let's go down here to our other tab. So to compare to skeletal muscle, we have still these striations, but now this pattern is a lot more regular. There's no branching of the muscle cells. And so what we're seeing is each of these, basically these patterns here, running longitudinally, each of those is going to be a muscle fiber. So let's go back to our protein atlas now. We have one more type of muscle to look at, and that is going to be our smooth muscle. And so instead of going just to smooth muscle, we're going to look at the stomach of the GI tract, because this is one of those areas where we would expect to find several layers of smooth muscle. So at a distance, this is what our GI tract will look like. To orient ourselves, we have up top, superiorly, we have our epithelium. This is sometimes referred to as a mucosa, as we will learn in future labs. Let's orient ourselves to our basement membrane. So that's going to be this wave pattern we see here, dividing the epithelium from the underlying connective tissue. And we have a couple more layers that we're not going to talk about right now. So let's get into our muscle layers. So those will be deeper. And we will find that we have 3 muscle layers in the GI tract within the stomach region. So let's go ahead and see if I can click on that. So more superiorly, we're going to have an oblique layer. And these are all based on the orientation of the muscle, of the smooth muscle fibers. So we've got an oblique layer, a circular layer, and then a longitudinal layer. And this is all smooth muscle. So we can't voluntarily contract our stomach. That would be weird. So instead, we have this involuntary contraction to help aid the passage of food through our GI tract. A couple things to note about smooth muscle in general is it's typically mononucleate, so we only have one nucleus per muscle fiber. There aren't any striations, so this one is kind of identified based on the lack of those features. All right, so this should hopefully be a review for you. Let's go back to our notes now. And so let's get a little more into skeletal muscle, and we'll talk about some of these ways that we identify muscles. So we're going to start with this big picture. We'll get a little more specific and talk about some of the weird stuff we have going on with muscle histology. And then I just want to end with some general information about the muscular system. So the main ways we can identify muscles is by anatomical terms like directional or regional terms. We can also look at things like the number of heads. So a lot of times the name of a muscle will clue us in to how many muscle heads there are within that muscle. One great example would be quadriceps. So the quadriceps tells us there are 4 muscle heads, 4 muscles, so to speak, within the quadriceps muscle group. And then these last 2 are starred here, fiber orientation and action, because these are some of the most common ways that we will see muscles be named. And if you go on the cadaver lab, this is going to be the exact same process you use to identify muscles, but I would always start with looking at the fiber orientation. So if we go down to our image here, what I mean by that is if we zoom in, we can see all of these muscles here have different patterns of muscle fibers. So in lecture, you're going to hear the term parallel, convergent, pennate, fusiform, all of these different types of muscles. These won't be tested in lab, but you can use this information to help you identify muscle actions or muscle names even. So let's start with a basic example. We have our rectus abdominis, and if you can see in this image here, we have muscle fibers going vertically in a vertical orientation. We've got a couple other things here, so we'll talk about these tendinous sheaths, but that's in a future lab. But for right now, let's focus on this vertical orientation. Think about what will happen when the muscle contracts. So generally when muscle contracts, it's going to shorten. These muscle fibers are going to shorten. And if we think about where our rectus abdominis is, this is going to cause our torso to flex, like during a sit-up. That is exactly how you can use fiber orientation to identify different muscle actions. So if you ever forget on a lab exam what the action of a muscle is, try to look at fiber orientation to clue you in. Let's look at another example here. So pectoralis major, it's our main muscle in the pec or chest group. We've got kind of this convergent pattern going on where we've got fibers splaying out and they're all converging onto one common attachment point. And so this muscle is mainly going to help with things like a chest press. Think about that motion where you're trying to flex your arms. You've also got a little bit of medial rotation going on. So based on how these muscle fibers are oriented, if you think about what's going to happen when they shorten towards each other, basically the fibers are going to go towards the more secure attachment point. So pectoralis major is going to have a lot of attachments to our thoracic cage. So what happens when we contract this muscle is it's going to help bring our arm towards that more secure attachment point. So medial rotation, got a little bit of flexion going on as well. So that's a little more of a complicated example, and I do ask you in your attendance sheet to practice with this. A more complicated example would be something like a circular muscle pattern. So we have a couple sphincters throughout the body. In this case, this is our avicularis oris, which is a muscle that surrounds our mouth and helps us to close our lips, pucker them, things like that. And so based on this circular pattern we see in the muscle fiber, that makes sense. Think about what will happen when we contract this muscle. It's going to help us to close our lips. And so this muscle is going to kind of contract towards the radial center of that circle to close the lips. So I think that's probably enough practice. We can move on, but that's what I mean by using fiber orientation to help figure out the actions and the names of muscles. So you'll have plenty of practice with that in your attendance sheet. All right, so let's go on to muscle organization. So these are terms that you want to be familiar with, commit them to memory for sure, because we're going to see this pattern, epi, peri, and endo, over and over again in all the organ systems that we talk about. So epi means above. This is going to be kind of that outermost layer that we see. Peri, I believe it means through. So this is kind of our intermediate layer. It's in between the other layers. And then endo means within. A little shaky on my Latin, but endo is within, so that's going to be our inner layer. And this is not unique to muscles. We'll see it with nerves. We're going to see it with anytime we have connective tissue surrounding an organ system. And so this refers to the kind of the connective tissue surrounding our muscles. We talked a little bit about that, but we've got our epimesium, perimesium, and endomesium. So we're going to take these prefixes and smash them onto mesium, referring specifically to the muscular system. So our epimesium is going to be that connective tissue layer that surrounds the entire muscle. Perimesium is going to surround individual fascicles. So let's start writing these out here. So the largest scale we will deal with is individual muscles. Perimesium is going to surround individual fascicles. I spelled that wrong for sure. Okay, let's try this again. And then our endomesium is going to surround individual muscle fibers. And one important thing to know is that a fiber in this case is the same as a cell. So anytime we use the term muscle fiber, that means muscle cell. So let's scroll down here. This is an example of one muscle fiber. So we're talking about one muscle cell. So we still have all the characteristics we would expect to find in a basic cell. We've got things like mitochondria. We have multiple nuclei usually scattered throughout our skeletal muscle. We will still have a form of cell membrane. In this case, we call it the sarcoplasm. So sarco is a term specific to the muscle system. And then this is not featured here, and it's something good to know, but it's not going to be tested in a lab setting. We also have this term called sarcolemma. So sarcolemma is going to be our membrane, and then sarcoplasm is kind of the inner part of our muscle cell. Kind of like cytoplasm in a regular cell. And then continuing the pattern, we have a sarcoplasmic reticulum. So this is basically a web, a network surrounding and permeating through the muscle cell, and it stores calcium, which is very important for muscle contractions. We'll talk about that in just a second. But this is basically going to be a sponge for calcium. So anytime we're not using that to contract our muscles, it's going to be stored in our sarcoplasmic reticulum. So that's pretty much all you need to know about muscle cells. Is we've got our sarcoplasm containing everything in the cell. Sarcolemma is going to be that plasma membrane. And then sarcoplasmic reticulum is going to be that inner network for storing and releasing calcium. Now, to go back to our connective tissue, one big concept I want you to know is, and remember, is that this connective tissue is not inert. It is alive, it's innervated, so you don't need to know these cells, but just be aware that there are going to be a ton of different cells within that connective tissue. A lot of people think of it just as like a passive material, holding things together, but it does usually have an immune function. We've got adipose tissue, so it can help to store energy. It's also helping to regulate tension between muscles. So we've got basically this really complicated network of connective tissue that is alive, it is innervated, it's supplied by blood vessels that is acting throughout the body.